share my I may still talk a little bit strangely because I had COVID, but it's now all fine. So rule-based modeling is pretty like already more than 10 years old, and in Virtual Cell we implemented it several years ago. Not all of you probably aware of it. So in a nutshell, if you have components like molecules in your models that have multiple states, say for example PLC gamma can have multiple tyrosines that can be phosphorylated, unphosphorylated, can be active, inactive. If you have complexes that consist of multiple molecules, if you have multi-site molecules, for example, here are some typical examples of like NWASP and NIC, but in this case they are generic molecule consisting of 4 sites, and then B consists of another 4 sites that can all interact between each other. Then these are types of models that can be tackled with rule-based modeling very easily. And by the way, the last model, directly Virtual Cell was used to prototype the models that later were simulated with Spring Salad. And this is cartoon of rule-based description designed in Virtual Cell that was later implemented in Spring Salad. And the difference is that in Spring Salad, you have volumes and you have distances between sites, while in rule-based modeling that is part of Virtual Cell, you have just components of molecules. By the way, feel free to interrupt me at any point. So now, completely in a nutshell. So I will have 2 slides and then I will open it for questions. So 2 rules in Virtual Cell would be written like that. It is a graphical user interface. So we have a molecule A that has 2 sites, B and C. We have a molecule B that has 2 sites, A and C. And we have a molecule C that has 2 sites, A and B. And we have 2 rules that A can bind to B and B can bind to C. So A, B, we have a complex, B, C, we have a complex. Then there are 2 ways to, and this is very simple description of a model. Then there are 2 ways in Virtual Cell to deal with these 2 rules. First is network generation, when these 2 rules actually lead to 1, 2, 3, 4, 5, 6, 7, 8 irreversible reactions. So it's A times 8 plus B, then it's A plus BC, because we have a BC formed here, B plus C. It's A plus BC goes to ABC, and it's C plus AB goes to ABC. So we have, out of 2 rules, we have 4 reversible reactions. And this is what Virtual Cell does for you. Based on the set of rules, it generates the set of individual reactions that later on can be used in regular Virtual Cell modeling. It can be done in as ODEs, it can be solved as stochastics, it can be placed in different types of geometries, and even run with spatial stochastics. So this is one way. And the second way,
Michael, could I just break in? There's a question in the chat for rule-based modeling. Is there a physiology reaction diagram or no?
Yeah.
And maybe you could just explain real quickly.
So let me Stop sharing, and let me reshare my screen. And on my screen, where is my virtual cell? Yeah. Yeah, so here is a specification of a model as a set of rules. When you look to reaction, the list of reactions, this is the way how you actually should do it, because it's like with having multiple sites on a molecule, it's very difficult, if sometimes it's impossible to draw the cartoon. But then there is a representation of a cartoon, and you can track actually what's going on here in a cartoon. So you have two ways of representing. This is, I guess that you see this double. It's a graphical card of my laptop. So yeah, let's look at this representation. So you have a EGFR molecule. It can, let's see if it can, yeah. Okay, so this EGFR molecule can participate in multiple rules. So if it was unphosphorylated, it can become phosphorylated. If it was phosphorylated on another side, it becomes dephosphorylated. Then another phosphorylation reaction on another side, and it can bind to Schick. So in short, there is a way to visualize after you do rules, but that's what a very good question, and this is part of my like advanced features. Rules are complicated to deal with. It's very easy to start defining molecules like that. Everyone is, when I teach it for students, they're absolutely happy and start adding it. And then it's very good to specify species. Species is a specific, well-defined object. So if molecule of EGFR can be in multiple states, so it has two tyrosines that can be phosphorylated, unphosphorylated, and then species is the same object in one state, and then you can quickly do reactions. And this is example of reaction when EGF ligand binds to EGFR receptor, and so it can bind to, you see question marks, to any receptor, whether it's unphosphorylated, phosphorylated, bound to something else. So it's very easy to define rules like that. What will happen as a result is not clear, because you have to have very good understanding of how the model works. So this reaction diagram can help you, but what I always teach is that you never go like with more than 2-3 rules before checking everything what can be checked. And specifically, the model, so let me again jump to presentation. So here you have as simple system as it can be. It's just molecule A with two sites, A1 and A2. And a single possible rule that free site A2 and free site of A1 bind together. Guess what? You have this single rule generates infinite chains of polymers, because you bind A to A, then you bind another A here, you bind another A here, 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 here. So therefore, virtual cell has a lot of built-in features that prevent you from running or not prevent, but that inform you if something goes wrong. So you can limit number of iterations. So in this case, you add these molecules not more than 5 times. You can limit the number of molecules in species that have in virtual cell. So in this case, you should have not more than 10 molecules. So the largest species would have 10 molecules. You can limit the number of species that generated, because in this case, you just can go and limit. So you can limit the number of reactions. You can limit the number of specific types of molecules in a system. In this case, you may have not more than 100 A's, and then I have B and C in a model. So let me see what's another question. Is there still a physiology? Yeah, okay. So this is one way what I told you, like how you can use rules to generate model that is more or less a regular model you used to work with. Another way is is what is called network-free simulation. In a network-free simulation, you have the same rules, and generally virtual cell is built in a way that physiology can be used with almost all applications. So in this case, having physiology and having set of rules, you can run them either extending them to the whole set of reaction, or you can do network-free. And network-free means that you don't really extend, but you use stochastic simulations to start with molecules, like in this case, 100 molecules of ABC, and simulate the distribution. In this case, single run of network-free simulation will produce 70 species ABC, 13 species AB, 17 species A, and so on. And another run would produce similar distribution. And in this case, when the system is really small, NFSIM is not important, but let's see what will happen when we run into pretty small, relatively small system. This is tutorial rule-based GFR compartmental that has only 10 reactions. And in these 10 reactions, you have like you can follow a tutorial and say that this and see that this 10 reaction would generate 116 species, which is pretty big number. So let's see, specification network. So let's see how many species will be generated by these 10 reaction rules. And the point is that not all of these 116 species will actually exist in a model, or in this case, even 55 species, or it's even less. So out of this 55 species, if you look at NFSIM, network-free simulations, then only few exist in reality. So those are agents that exist. So although theoretically, what I said, the full expansion of the set of rules would produce 55 different species, in stochastic agent-based simulation run by network NFSIM, you will have only few selected species being populated at any given time. And when you do large models like clusters and what I said, polymers, then NFSIM is the only way to do, because you cannot generate the system that would include all polymers of any length. But if you have a system, if you can run it with NFSIM when model is keeping track only on real existing species, and basically you may have, say, a tail of this distribution each time in your model. So I will just mention few features, and then so observables, something that you have to keep track. Output species, what I just said. Anchors for spatial modeling, and anchors is very important because they will keep your species constrained in a given compartment. And then there are advanced features for NFSIM, advanced controls, that if you are really doing something serious, you have to go there and click on question mark, try to read it, and if not, try to ask us if it's not easy.
And also remember, you can use the help. So there is help on them available as well. So
okay, so this is all what I wanted quickly talk about.
So are there any other immediate questions from anybody else on anything? Kopasi, steady state?
Okay. I have a question that's like super general about the physiology of things. I guess, what if you say you have a protein that has been implicated in participating in certain signaling pathways, maybe through like co-immunoprecipitation experiments and things like that, but it's not exactly known like what binds to what exactly. If I were to model something like that, would that be a predictive model? And then that would be followed up with experiments. I've never done modeling before.
Yeah, so if you don't, already have a specific known reaction with reaction rates and things, you can basically test a particular pathway and see what it would generate in terms of species or observables, as Michael would call them, that you know of, that you can measure, right? So there are certain things that usually that you can measure, and you know how they change over time under a particular situation. And so you can then apply, make up some, almost as Padmini did, right? She just created a function that would show how a reaction mechanism changed as a function of that species. So you could use it as a catalyst in a way, or you could use it as being used up in a reaction or binding and also changing the system that way, and see how that affects your final outcome, right? So a model is a hypothesis. It's the same thing, basically, right? So, you draw your little arrows and boxes on paper. Well, you're doing the same thing. You're just adding some numbers along the way, right? And you can adjust the numbers to see if you can get something that fits what you observe. So one of the original papers we did with Visa was a really, that came from Les's lab, was a perfect example of creating a quantitative hypothesis as a model, and showing, first of all, that the model wouldn't fit the data. So the model had to change in order to make the model right. So in this case, it was whether the endoplasmic reticulum was evenly distributed in the cell or was concentrated more in the nucleus, right? So that had to happen in order to ever be able to fit the data that was known in the biology. And then there was another case in that same model where it wouldn't fit, and they realized that they'd taken some numbers of species that were actually done in a different experiment and not exactly the same cell type. So they decided maybe the data was wrong in this case, and then they redid the data and found, sure enough, that concentration was not right. and put that correct concentration in the model, and now it was predicted. So usually you kind of build a model to fit certain experimental situations, and then you ask if it's predictive, right? So you build the model, it fits your data. Now you do something new with that model, and that's an experiment that then you recreate the experiment in the laboratory. So it's a process, right? So it's perfectly fine to make something up. That's what you do in a hypothesis. But then you need to test it and show that it fits the experimental condition. Does that make sense? Is that what you're asking?
Yeah, because like I knew that some people will build a model and then do the experimental data and see how it matches up, and then other people will do the experimental stuff first and then build a model based off of that.
Right. But you're building the model off of somebody else's experiment, right? I mean, that's the way science works. Somebody did the experiment 5 years, 25 years ago to show that adenyl cyclase is important in some reaction. So it's not making it up. It's based on what's the known parts of the system, but then you reach something that you don't know. And we're not modeling from first principles. You're always making an assumption. So you're not modeling every molecule at every time. You have to you make some adjustments for reaction rates are simply expressions that define how things are known to behave, right? It's not, you're not at that atomistic level or anything. So you're already making some approximations along the way. It's just important to sort of understand what those approximations are.
Mm-hmm.
Yeah. So
does anybody else want to jump in?
Yeah. Meanwhile, I answered a question about how parameters can be, NFSIM can be run with multiple parameters. So that's how it's done. You basically, for any virtual cell model, you can do parameter scan, or you can manually define the range of parameters and run it on a server. And so this is done for any models, including very large models run with NFSIM. And you can go and check how the simulations are varied when parameters are different. In this case, so we have to talk about total phosphorylation. Yeah. So I varied phosphorylation rate, and I got now simulation results for different dephosphorylation rates. Okay, so hopefully it answers the question.
Yeah, you can do that with any virtual cell model. It doesn't have to be NFSIM, obviously.
Yeah. Then you can do.
Can you show how to do that? Like, where did you put the parameter values?
Yeah, so this is, once again, I can repeat it. So a lot is done through this edit simulation. And in this edit simulation, you can select some of the parameters and do scan. And then when you do scan, it gives you either the list of values which you can enter, or you can go from, say, 1 with 4, 5, and you can even do log scale. So it will go into 1 to 1.1.001 and so on. So, and then you just run it, and you run it with a green button, which is sent to the server. For stochastic simulations, there is another option which I'm not sure if it's available already in release version, but we can immediately vary the seed and so send multiple simulations with different seeds, so which will give you the averaging over multiple stochastic simulations. For NFSIM and for any ODE-based models, you just can vary do parameter scan.